Well, good afternoon. Thank you all so much for coming. It's a pleasure to have you with us today to dig into soil health. I'd like to thank everyone who provided feedback for our previous virtual field days. We really do appreciate you giving us that honest feedback. We're still working to perfect the technology side of things, still trying to get that good audio and video quality for you. And partly why we're doing these field videos is because it allows the speaker to cover or address additional items that they may not remember or think of when they're looking at still images and describing them. Since we're using this technology, we get to have the benefit of them being out in the field while we get to enjoy the comfort of being indoors. A little bit about the Iowa Learning Farms. I'm Liz Ripley. I'm the Conservation and Cover Crop Outreach Specialist. Our organization was established in 2004. Our goal is to build a culture of conservation by working with farmers and researchers to implement practices that improve water quality and soil health while remaining profitable. Our partners include the Iowa Department of Agriculture and Land Stewardship, Iowa State University Extension and Outreach, Leopold Center for Sustainable Agriculture, Iowa Natural Resources Conservation Service, the Iowa Department of Natural Resources, US EPA Section 319, Conservation Districts of Iowa, Iowa Farm Bureau, Practical Farmers of Iowa, GrowMark, Iowa Agriculture Water Alliance, Iowa Corn, and Iowa Nutrient Research Center. So just a quick couple reminders. We do ask that you mute during the video portion, but then we do invite you to unmute to ask your questions of Marshall today, or you can submit them via the chat box and I'll make sure that those get to Marshall. We will be sending you an evaluation, as I mentioned at the start of the event. This will come via email this afternoon, so please be on the lookout for that and get that submitted. But this is just the start of this process. So it's the start of the conversation. We're exchanging ideas. And then it's also important to network this with those that you interact with, either as a technical service provider or as a farmer and landowner, to share these ideas with others and return that evaluation for us. The good news is that these are also recorded so that we can post them online. For folks that weren't able to attend live, you can certainly share the link with them. Today, we're excited to have Dr. Marshall McDaniel, Assistant Professor in Soil-Plant Interactions at Iowa State University, who's going to be sharing with us some of the work that his group is doing. So while I'm getting ready for the next slide here, Marshall, if you want to say a few words while I get the video started.
Good afternoon, everyone. Liz, thank you for inviting me. I'm happy to be here, and I'm excited to talk to you all about soil health. My name is Marshall McDaniel, as Liz said, and I'm a assistant professor in agronomy at Iowa State. And I think that's all I have to say for now. I look forward to fielding your questions, and I'll be ready after each video. Good morning. My name is Marshall McDaniel. I'm an assistant professor in the agronomy department at Iowa State University. We're out here near the Biocentury farm that's right behind me, and we're out here to talk about soil health. Okay, so right here we're in the middle of a long-term experiment. It's called the Marsden Farm Experiment. The primary investigator here is Matt Liebman. He's also in the agronomy department. I work with him. And what this experiment is testing is the long-term effects of diversified crop rotations. And there are many researchers besides me that work on it. I happen to be looking at the effects on soil health, but there are people looking at weed suppression, water quality, carbon sequestration, the economics of 4-year rotations versus a 2-year rotation. And this experiment was established in 2002, and it has 3 treatments. So there's the kind of business as usual corn-soybean rotation that receives synthetic fertilizer based on soil tests. And we have an example of one of those plots behind me here. Right now there's corn planted in this plot that's to my right behind me. And then there are 2 other treatments. There's a corn, soybean, and then oat, red clover rotation, and that's rotated every 3 years. And then there's also a 4-year rotation that's corn, soybeans, oat, alfalfa in 1 year, oat as the companion crop, and then alfalfa in the 4th year, and then it goes back to corn. And so we know that there are a lot of benefits to yield, both the cash crops that we're looking at, but then also there are other benefits to soil health that we're trying to investigate in my research group. Soil health is all the rage these days. Everyone's talking about it, from researchers to farmers. And to me, one of the basics of soil health is monitoring change after a management practice. That's kind of the idea and underlying principle with the soil health movement and what people are so excited about. And there are a bunch of ways that we measure change in soils, and I'm going to talk about those today. The other thing that differentiates soil health from the terms before it, like soil quality and soil tilth, is the emphasis on soil biology, which you're going to hear a lot about today as well. And the underlying idea with that is that soils are a living, breathing organism almost, and we'll go over all the organisms that are in there and how they contribute to all the things we care about as humans, and how they can contribute to one of those things, which is growing productive crops and promoting conservation practices. All right, so soil health is defined as the continued capacity for soils to support animals, plants, and humans. And within that, we kind of think of soils as being able to perform beneficial services to humans, and we call these actually soil ecosystem services. So many of these you might be familiar with, so just providing and provisioning food, fiber, and fuel, so growing crops, what is why we're out here. That's one of the main ones that agronomists are thinking about, but also sequestering carbon, retaining water, providing a habitat for soil organisms, a structure or a base or foundation for buildings to be built on. Those are all the things that soils do for humans, and we call those soil ecosystem services. And I'm going to talk more about those today in more detail. But the main thing I want to talk about is that there are soil health tests out there, and many of these tests are performed by laboratories. So you can grab a soil sample from a field and send it to a laboratory, and they will give you back an analyses that's similar to a soil fertility test. But one of the main things that differentiates these commercial soil health tests from soil fertility tests is that they give you back maybe a score in many cases, and they're also measuring some aspect of soil biology. That's the thing that really drives them. And they also will charge you a little bit more for those. They're usually more expensive than just your basic soil fertility suite that gives you NPK and your other macro and micronutrients. And we're going to talk about those today.
All right, so now it's your time to submit your questions or ask those of Marshall, and I'm gonna have you turn your camera on too, Marshall, and
Sure.
continue the discussion.
Here, let me put my mask on too. No, I don't need to use this in my office. Yeah, we didn't go into a lot of details. It wouldn't surprise me if there aren't very many questions yet. But be thinking of them as we go through the next few questions or videos.
So Marshall, what are some folks, or what are some things folks can look for that might be an indication of soil health from a visual standpoint? Are there any things that can help them that way?
Yeah, there are a lot of, and I like to call these just using your senses soil health tests. And I've worked with a lot of farmers that give me anecdotal evidence for this as well. You know, being able to get out into a field earlier because field is drier, usually meaning infiltration is faster of water in the soil. And that was big for the last couple years where we had very wet springs and even wet falls. To get out into your field earlier makes a big difference. Other things you can look for, we've actually been monitoring another experiment that's nearby, the one that I was standing in front of in the video, but we've looked at the impact of restoring prairie to soils, and you can actually notice visually that the soil is just darker and there's more roots and in the soil after you sample it. But then also the aggregates sizes are larger. The soil aggregates are just the way the particles are sorted, those of you that aren't soil scientists. So the organic matter and all the inorganic weathered rock that's in the soil, usually bigger aggregates, the shapes of the aggregates can tell you a little bit about soil health. And if we have time later, I do have some pictures from that experiment too, if people are interested in seeing that, and you can actually visually see it with your eyes. The other thing that is pretty easy to look for is, and we'll go into this with the do-it-yourself soil health test. So in many cases, watching how fast residue decomposes is a good indicator of soil health too. So a more biologically active, healthy soil is going to decompose residue faster.
We've had a couple questions come in regards to the rotations.
Sure.
So a little bit, if you can explain the 4-year rotation a little bit to remind folks of what that is, and then how do you utilize the oat, clover, and alfalfa in the rotation? So what do you do with those?
Yeah, so the oat and alfalfa would be considered hay for animal. And I think in that video, I forgot to mention too, the other difference between those treatments. I did mention that corn soybean rotation receives all synthetic fertilizer, but the 3-year and the 4-year rotation receive 90% or more of its nitrogen from composted cattle manure. So there's only some supplemental synthetic fertilizer to it. So really what it's simulating is a longer rotation where hay like alfalfa can be grown for animals, and then the manure is put back into those plots. I guess the buzzword for this now is called integrated animal and crop or a livestock system, an integrated livestock system. That was a good question. I hope that explains it. The tillage is a little bit different between them too, and I didn't talk about that. If people are interested in hearing the nitty-gritty details behind it, I'm happy to send out a publication from Dr. Matt Liebman, who I said was one of my colleagues in the department.
So that was a great question. You just anticipated that someone submitted was about the tillage. And then this last one before we move to the next one is, were the oats, clover, or alfalfa terminated in the fall or spring ahead of the return to corn and soybeans?
I believe they were terminated in the fall, and I know that they are terminated with moldboard plow. So they would be harvested and then moldboard plowed, and then to address the other chisel plow, I think after corn and soy, I think it's fall chisel plow tillage. Maybe just 1 year. I have to look back at that, but I'm happy to send those details of that experiment out.
All right, so I'm going to go ahead and share my screen, play the next video, and then we'll come back and answer some more questions.
So as I mentioned before, I talked about commercial soil fertility tests, and those involve grabbing a soil sample. Usually we take a composite sample from about a 2 to 2.5 acre field or portions of a field, and we send it to a commercial lab. They send us back results. Those results are the nutrients extracted at that time and at that place that you collected that soil sample. So a good analogy to that is that it's a picture of what's going on in your soil over the year-round growing season. And If you want a more kind of full picture of what's going on in the nutrient dynamics and what the root is experiencing out in the field, I might recommend this, which these are called plant root simulators, is the product name made by Western Ag. And what these are their resin, they're ionic resin membranes that are just inside a plastic strip, like a garden stake. And I'll explain these strips a little bit here, these resin membranes. So this kind of semi-transparent orange portion of these has a membrane in it that is either positively or negatively charged. So this one that I have the plus on, it's positively charged, or it's negatively charged, so it picks up the positive cations. This one is negatively charged and picks up those anions in your soil like nitrate. That's important. So how these differ than just grabbing a soil sample and sending it to a soil testing lab is that you're getting almost a movie instead of a picture, right? So I mentioned that a soil test is a snapshot of what's going on. These, you take a soil knife, and I'm going to demonstrate this in a moment. You place it in the soil so that it's under the surface of the soil, and then you press the soil back up against it, and it's collecting nutrients over however long you leave it out. From usually they suggest 2 weeks to a month, and then these are collecting nutrients as they're released from the soil, and however long you leave it out there, it's measuring the soil nutrient supplying power. So ammonium, nitrate, so your major forms of nitrogen that's available to plants, and then all your other macro and micronutrients are picked up with these two ion exchange resin membranes called plant root simulators, because they pick up kind of a more of a fuller picture of what a root is picking up. So I don't think we need to get out too far, but this is probably good enough. So I've taken the anion one, and we usually pick a row. Again, these are supposed to represent plant roots. So there are, for research purposes, there are many different ways to do this. So sometimes, you know, we'll put something around maybe a PVC tube to exclude roots. But here, and what we often do in my research group is try to look at what the root is actually seeing. So I'm just going to take a soil knife and kind of create a nice crevice. I just put the soil knife all the way through the surface of the soil in between the soybean plants here, and then I take the soil probe and I put it up until about, I have an inch above the surface. And you want to be consistent. If you're looking at any treatment effect or anything, the important thing is being consistent, and I usually leave them about the top of this about an inch out of the soil surface. And so that's what it looks like. And then to finish it off, I usually just take another stab at the soil and push the soil up against it so you have good contact. And over here, do the same thing. And so now you've got soil that's in good contact with the probe, and you leave it out. So in about 2 weeks, we'd come out and collect this.
All right, so we've had a couple questions come in.
Sure.
The first one is, what is the thought on advertised biological enhancers, biology in a bottle, due to the residue buildup is it indicative of compromised soil health?
Those are two great questions, especially the bugs in a bottle. I get that question a lot. And I think as a scientist, and many farmers and growers are also, I think like scientists, we're skeptical unless we see the evidence that they work. But you know, I have seen some evidence. We've got a student who's working on a project looking at biologicals, and they seem to work in some cases. And my previous philosophy, and so this is a good example of people changing their minds once they see the evidence. So I think there's some potential. I think it's one of the frontiers of soil science. We really don't understand it. But one of the things to always think about, and I'm not a soil microbiologist, I play one on TV a lot of times, but that there are so many microbes living in the soil, in and on our bodies as well. So we can use the analogy for our bodies that when you add a organism that's been pampered in usually at the appropriate pH, you know, at the optimum temperature, and then we throw them into a harsh world, whether it's a human gut or the soil that is a lot drier, a lot harsher, more competition, it makes it a very difficult situation for them to outcompete and do well and thus benefit soil health. But that's a great question. I get that question all the time. So I'd say there's potential maybe, but stay skeptical. With the what was the second question again?
So is it indicative of compromised soil health? So
yeah, that's another good question, and this is something that I think is on a lot of people's minds, especially with a lot of the companies out there that are getting into the carbon markets, like Nori and Indigo Ag. So, you know, while residue accumulating on the surface it can be a good thing in many cases, right? It keeps the soil cooler. If it's not mixing with the soil, there's not a lot of biological activity to recycle those nutrients back to the soil. There are some cases where too much residue on a soil can be harmful, especially if it's not being recycled, and it can cause the soils being too wet, right? This is a problem that people have, and why a lot of folks decide to till, because incorporating residue in the soil usually increases its decomposition or cycling of nutrients, and use of that carbon that's in the residue for organisms in the soil to decompose and use as well.
So you kind of led into this one, but what is being done regarding carbon sequestration? You mentioned the markets there.
Yeah, well, how much time do you have? Yeah, so that I mentioned two companies, and there are others out there in this space too. And I did a little bit of work in Australia before coming back to the Midwest. I lived in Australia for 2 years, and they instituted briefly, and this was before I got there, a carbon trading system. And I know there's a lot of overlap with this carbon talk and soil health, because I think that is a good indicator. I think it's not the only indicator to look for, but a soil that's increasing carbon is more or less likely, in my book, described as a healthy soil too. There's some caveats to that, and we shouldn't myopically focus just on carbon. But yeah, I've been on some Indigo Ag panels with growers and other scientists and agronomists, and there's a lot of interest in it. I think it's a potential where farmers can be getting paid for something that they're doing already, and also promote soil health. People use the phrase win-win a lot, and that's thrown around quite a bit, but I think it's something that's interesting that you all should be looking out for in the near future.
I've got one more carbon question, then a couple about the indicators in particular. Can you explain for those that maybe don't understand or know how plants sequester carbon and how it's measured?
Yeah, this is another one that I'll try to condense down. But so plants basically take CO2 from the atmosphere and then convert it into their tissue, right? So there are whole classes in our department that are taught on this, and I'm not the one who teaches it. But then that plant matter can either be in the form of roots or the above ground stuff that we typically see, right? We don't see a lot of what's going on below ground. So the plant is sequestering the carbon from the atmosphere in its tissue there, but there's also a counteracting force of decomposition, right? So there are biological organisms in the soil that are like us. They need a source of sugar or carbon, and they use oxygen, and they create CO2 out of that. So it's kind of a balance from the CO2 that the plant's uptaking versus the CO2 that's released from residue decomposition. And one thing I haven't even touched on yet, but is a really cool area of research, is there's a lot of carbon that comes from the atmosphere through the plant tissue and then directly out into the roots. That's not even a part of the root tissue. So this we call root exudates. Some of you might have heard this term before, or rhizodeposits. These are forms of carbon that we can't even measure in a plant really without special equipment. And it's constantly, a plant's like a straw. It's taking CO2 from the atmosphere and sending it out through its roots to the soils. That's a great question, whomever asked that one.
So a couple specific to the indicators. One, how expensive are they? Is there a time when they should be out there in the growing season? And should this be replicated based on soil types and crop types?
So I think that's going to segue nicely to the next video, but I'll get a start on that. I'm actually going to share my screen. Somebody asked how, that must have been a question from Mark Licht. I think I planted a question with him. Let me share my screen here. All right, so can you see this, Liz? I know you're the only one that can talk to me.
Can
you see a table?
No.
No, you can't? How about now?
There we go.
Okay, great. Yeah, so this is a table that I use in a lot of my extension talks, and it's just for organizations that do soil health packages. So I mentioned how there's kind of a difference between like the traditional soil fertility suite that you all are probably familiar with. And then these are labs that have a basic soil health package and then kind of add-ons to that. And you can see the range in price, if I could get this to go. So they range as low. And again, this is just selected for labs. These are very common ones we use as researchers, and I hear growers use as well. So they range from $45 to $170 per sample with an average of $78. And, you know, I don't have to be an accountant to know that this is quite a bit of money, right? And especially for times now where margins are very thin and productivity on farms. And so one of the areas of my lab research, and why I kind of alluded to Mark maybe planting this question, one of the areas my lab group does research in is looking at do-it-yourself or DIY inexpensive ways to measure soil health, besides what I had talked about before with using your senses, visually seeing it, noticing that you don't have to use as much downforce in your equipment. So things like that. If you want to be more quantitative with it, Jack Boyer's on the call here. He's worked with us in a study where we were measuring decomposition of tea bags in soils. I worked with some other farmers where we found some interesting results, and I think that's our next video. So maybe we should go to the next video unless we have time to have another question.
We'll come back to a few more of these questions, but let's go ahead and move on to the next video. Oops, make sure I'm sharing the right screen here.
So we just talked about a different way, as opposed to traditional commercial soil tests, to measure nutrient supplying power, which is a soil ecosystem service. Another soil ecosystem service that we kind of attribute to soil health is leaching of nutrients. And so we actually want to leach less nutrients. A healthy soil should supply plenty of nutrients to a growing plant, but leach very little of it. And in my research lab, we use another kind of simple technology that uses ion exchange resin, but beads instead of membranes. So the way this works is, you can see this is PVC. You could buy most of this at your local Lowe's or Tyson's or Menards. It's just a PVC coupling with two pieces of 2.5-inch PVC and or 2-inch PVC and nylon mesh holding beads in this nylon mesh. And just like those ion exchange resin membranes that are on the plant root simulators, these ion exchange beads are both they're positively and negatively charged beads. And what those beads do is they collect both the anions and cations that are leached through a soil profile when we bury this in the soil. So the way this works is we either have to come out with a shovel or we have to come out with a backhoe and somehow dig a soil pit. And then on the wall of the soil pit, we dig a tunnel that goes under the soil surface. So we're kind of under a profile of undisturbed soil. And then we bury this resin lysimeter, and we backfill that side tunnel and then fill the main tunnel. And then we leave these out typically a whole year. And over that year, soil water's infiltrating and then leaching through the soil profile and through these resin beads that are collecting both cation and anions. There are other ways to measure how much nutrients are in the soil water or being moved through soil water. And one of those are what's called a suction lysimeter. And there's one of these at the edge of this plot here. And this has just a cap on it. And then there's some corrugated plastic. And then inside here, there's tubing. And this tubing, we put a vacuum on. And then 24 hours later, we can come out and collect soil water from a porous cup buried in this field at about 4 feet deep. And that tells us, how much of the nutrients that the crops aren't picking up, but that's being leached potentially to groundwater and then maybe to surface water. So this is how we tie soil health into water quality.
I'm sorry about that. I thought that was the video where we showed the tea bags, but I think that's the next video.
So there was make sure I'm unmuted. There was a question that came in. How much money could producers potentially save by utilizing some of these tests?
Well, I haven't done this calculation, but I mean, I think it's not just about the money that you could save. I think it's more about overall productivity, and maybe the money you save not on gains in yield, but also reductions in input. So a lot of the farmers that I work with or I talk to, they do this a lot for the reduction in inputs, so that nutrient supplying power. I'm not aware of anyone who's done a economic calculation on kind of the benefits, the financial return on soil health, but there are folks that are looking at one aspect of it. For example, the nitrogen supplying power. There is now 2 studies that have looked at what's called and I think we can talk more about this too if there's more questions on this. One of the more famous soil health tests is called the Haney test, and it's a CO2 burst. And what happens is as that CO2 is released, it's a measure of microbial activity in the soil, and it also relates to the nitrogen supplying power of the soil. So a couple studies that came out in 2018 showed that the greater that CO2 burst, i.e. the nitrogen release from the soil, the less nitrogen that corn crop needed to get to the optimum yield, so 100% yield. So another way to say that is that the soil is supplying more of the nitrogen and you have to put less fertilizer on. So that's one angle where soil health directly relates to your pocketbook.
And this may vary by soil types and things like that, but is there an optimum for like nitrogen or some of these other measurements that the labs are conducting?
An optimal for soil health?
Yeah, that people should be aiming for when they're taking their
and
sample.
actually this is where a really cool area where kind of old fertility tests meets new soil health tests. So some of my colleagues in the agronomy department here, Mike Castellano, John Sawyer, and Mike's graduate student that's now at University of Kentucky, Hannah Poffenbarger, did a study to look at carbon sequestration in continuous corn. And she found that the kind of point of greatest carbon in the soil also happened to correspond to the agronomic optimum nitrogen rate. So going back to those old agronomic optimum nitrogen curves and relating it to soil health. So if you're applying nitrogen at what the crop needs, you're also probably benefiting your soil the most as well.
So if someone were to maybe try to make that PVC resin membrane, where would they find those resin beads?
Send me an email, but in short, the membrane, so the PRS probes, the company is Western Ag. They are on more, so we talked about kind of cost efficient ways, and we'll talk more about this. They are rather on the expensive side compared to some of the other DIY soil health tests that my lab group has been working on.
Hey, Marshall, this is Jackie Comito. So some of us can actually talk in and pipe in and talk to you if we want to.
Okay,
so just
great.
let other people know they can. To me, I want to go back to that analogy you used that I thought was really powerful, the difference between a still photograph and a motion picture. In other words, you're getting a sense of time too, what's happening over time. And am I understanding that correctly, that things like the Haney test aren't really giving you the sense of time that these DIYs are giving you?
No, the Haney test is a measurement at a point in time. However, it's measuring a process rather than just a pool of nutrients that's available to the plant at that time. So in other words, it gets closer to the analogy of the movie. It tells you maybe how the story would unfold for that year. Because you're measuring something that's a biological process, in other words. And yeah, as Jackie, I don't think I went over this in the video, but I talk about this a lot at extension talks I give. So yeah, those like the late spring soil nitrate test, that's just a snapshot, right? It's just a pool of nutrients, but there are ways to kind of uncover or to view the full movie instead of just a snapshot or photo. And there are three ways to do that. One is to measure a biological process, as Jackie kind of alluded to with the Haney test that has related to nitrogen supplying power. Another way is to measure a form of organic nitrogen that we know is kind of made available by microbes to the crop over time. And there was a test that came out of University of Illinois that did this a couple decades back. And more recently, Steve Coleman at Ohio State University is working on, or has worked on a test that can measure this organic form of nitrogen that can be released over the growing season. And then a third way to do it is with that resin membrane that I had showed that you can bury out in the field and measure the nutrient supplying power that is integrating over time, like 2 weeks to a month.
All right, now we are to the DIY Truly video. So I'm going to go ahead and share that screen and we'll take a look.
We can use a different varieties of tea, and it's good to know what type of tea. Some tea actually decomposes a little faster than others. This is a rooibos or red tea, and it has a higher carbon to nitrogen ratio. The higher the carbon to nitrogen ratio is, the slower the tea will typically decompose. And we think that soil that's really healthy should decompose something that's a little more tough on average to break down. It'll decompose it even faster. So what we do is we come out and usually about a third of the way in between two corn rows, we just dig a a small hole about 3 inches deep. We place the tea bag into the hole and then just gently bury it. So after we bury this, we can mark it with a flag and come out and retrieve it in about 2 weeks. Is enough time for decomposition to occur. With green tea, which has a lower carbon to nitrogen ratio, decomposes a little faster. You might want to get out here in about 4 or 7 days, so just about a week.
So how many sites have you utilized this method on already, Marshall?
This is where I'll put in a plug for the Leopold Center for Sustainable Ag. It was the first grant I received when I got here to Iowa State. They funded us for a 2-year project. In the first year, we worked with Practical Farmers of Iowa. I'll also put in a plug for Practical Farmers of Iowa. I think there were a total of 9 or 10 farmers. Jack Boyer, who's on now, was one of the farms. There might be some other ones on the webinar that participated as well. And we were basically comparing cover versus no cover. And some of those farmers had cover crops in place longer than others. And I should mention that they were strip trials. So they had replications on their field of at least 3, but sometimes even 4 or 5 strips of cover versus no cover. And so that was in 2017. I have a student working on publishing that data now, but I'm happy to send some preliminary results. And then in 2018, we took a different approach and instead of trying to cover a lot of the state of Iowa, we focused on long-term sites that are just in central Iowa on the same type of soils. So we buried tea in those plots you just saw the video of me at. So, you know, comparing conventional versus diversified rotation. We buried tea under 12 years of restored prairie versus corn and soybean rotation. And various other treatments, even adding biochar, residue removal. And we have some interesting results from those that my student is working on writing up and publishing right now.
So we had a couple questions come in about specifically measuring the decomposition, whether it's on visual inspection or weighing them, and how much decomposition is telling you what in regards to their soil.
Yeah, so to measure it, all you need is a 2-place balance. And those can range in price. Usually just like an automobile, the more you pay, the better it's gonna be. So the more accurate, more precise. But you can get them as inexpensively as, I think last time I checked, Amazon has them for $40, $50, a 2-place balance. So you need some good precision on there to notice a difference, because the tea bags themselves are only about maybe a gram and a half, 2 grams. So the idea is you weigh it before on that scale, you know, take some notes. Then you go out in your field, you bury it, and then after, like I said, about a week or 2 weeks, depending on the tea, you unbury it and try to clean off as much soil as you can and reweigh it. And the mass loss, or the amount of tea that was decomposed, is a good indicator of a healthy soil. So the healthier soil, more biological activity, greater nutrient cycling, the faster that tea is going to decompose. And in fact, the red tea seems to be a rooibos tea, which isn't very easy to find around the Midwest, but the black tea will do the same thing. It has a very high carbon to nitrogen ratio, similar to corn residue.
And so they would take that number divided by the original, and then what does that tell them? in terms of?
That's just the amount of T that was lost.
Okay.
So you subtract the initial versus minus the final, and then some people express it as a percentage. So then divide it by the original weight too, and that's the percent of T that was lost from that tea bag. And I don't want to dive into the details on like dive into the weeds on this, but if anyone has more specific questions, we have a protocol that we wrote up for the PFI farmers like Jack that's on the call with us now that I'd be happy to send to folks.
That would be great. Yeah, we can post it on our website after this and make that available alongside the link for the video. So in comparing healthy and less healthy soils, will the microbes vary more in quantity or type?
Oh, that's another great question. How long do you all have? So we know that besides quantity, I think is easier to measure than type. You need to do, there are commercial labs that will tell you type. So most, the commercial labs I've seen, they use a biochemical markers to tell you, how many gram-positive bacteria, how many fungi are in your soil. So it gives you more of an idea how the type is changing after a management practice. There's even more kind of sophisticated, and usually with sophistication comes money as well. So there's usually even more ways to kind of look into the microbial community in your soil by using DNA fingerprinting. And this is usually reserved, at least right now, to mostly researchers, besides the biomarker test I just mentioned. But it's rather easy, and we do this in my lab as well, just to measure the quantity of living organisms, or at least microbes in the soil. And the way we do it, and this method's been around since at least the 1970s, 1980s, is we actually use chloroform, which isn't very safe to use, so I don't recommend doing this at home. But I think there are 1 or 2 commercial labs that do this as well. So they'd take your soil sample, they'd split it into 2. One would get gassed with chloroform, so that's the the knockout gas you see in the movies, and that what that does is it kills all the organisms in the soil, and then you can extract their bodies and what was inside them. And then you have a control soil that wasn't gassed, and you do the same extraction. And the difference between the killed and non-killed is the amount of microbial biomass you have. And I could actually show some data from that microbial biomass from those, the Marsden plots, the rotations there if we have time.
So I'm going to go ahead and play the last video, and then we'll convene back for more questions.
Okay.
One of the keystones to soil health is the living organisms in the soil that drive a lot of the soil ecosystem services, or the benefits that human beings and plants and animals derive out of soils. And one of the main findings that one of my graduate students found was that the microbial biomass, or just the mass of the amount of biology under the 4-year rotation, which is to my left, compared to the 2-year corn-soybean rotation, is at least 25% greater, and that's consistent year-round. Almost the entire growing season, there's more microbial biomass under these soils, under the diverse 4-year rotation, compared to the 2-year rotation.
All right, so that kind of segued well to one of the questions that had come in.
That was perfect. Almost seems planted.
Yeah. Is there a way to achieve maximum nitrogen availability under one of these particular crop rotations rather than using commercial fertilizer?
Yeah, I think that rotation is a good example of that. The biological activity is much higher, thus nutrients are cycling more quickly over the growing season. And Yeah, the yield data from that 17, 18 years of that experience kind of bore that out as well. So I can't remember how much the corn yield is on average. I want to say about 6% higher in the 4-year average when you're looking at the corn phase of that rotation compared to the corn and soybean. But the soybean phase is even greater. I think the soybean is about 15 to 20% greater on average across the 17 years of the experiment.
What were the microbial biomass results? Were they similar in the 3-year rotation or closer to the 4-year or the 2-year?
Oh, that's a good question. And unfortunately, I cannot answer that, but I am going to show my screen again. So Liz, please tell me, since I've got you on here and I can talk to you. Can you see my screen?
Yes.
So I don't have any results from the 3-year. We only measured microbial biomass, and I actually underestimated it from my video. That 20 or whatever I said, 25%, that was just in 1 year. But if we look out, so this is the diversified crop rotations increased microbes by 62%, or the microbial biomass compared to the corn soy. So the corn soy is these circles with the dashed line in red. And then the 4-year rotation is the blue here. I think I could draw on my screen too if I need to. And we measured it between April and about November every year for 3 years of the rotation. And actually we're measuring it this year too. So we'll have the full 4-year rotation. And so you could see the phase that the 2-year was in: corn, soybean, corn. The 4-year was under corn, so that we synchronized at least the first 2 years of the annual crops here, corn and soybeans. And regardless of what crop phase you were in, there was an increase, even a greater increase in soil microbial biomass in the soybean year compared to the corn. And then of course, you know, you have different crops. So oat alfalfa is quite higher than the corn soybean as well. And this is just soil microbial biomass. So it was the data collected from that method I had mentioned, where you fumigate with chloroform one portion of the soil and leave the other unfumigated. And this is in a laboratory. We don't do this out in the field where under unsafe conditions. So yeah, this is pretty striking results. And one other thing I wanted to show, some preliminary results we have from this experiment, is the gravimetric water content too. So not only is the biology change, and this is actually getting back to something we were talking about earlier in the webinar, is some of these physical properties of soil changing, the infiltration rate, water holding capacity. So in the 4-year rotation, so on average, that 4-year rotation increased the amount of water that was in the soil by 16%. And this is really consistent all year round. So those of you that remember 2017 was very dry, so that you can see that in this dip. Gravimetric water content is just the grams of water per kilogram of soil in this graph. And we see the same thing in the soybean year. So, you know, more water. This especially helps, you know, when you're getting out to June, July, and August, when the crop is more water limited. And then the last year, 2019, we found the same thing, even greater differences. But this is probably driven by the crop is different in this last year.
So I've got about 3 more questions before we wrap up today. Would you expect these changes to be different if the plots were managed in a no-till system?
Yeah, there'd be some differences with no-till as well. And we've done, in fact, I forgot to mention, that was one of the treatments where we buried the tea bags in, and we found long-term no-till. So Dr. Mahdi Al-Casey is one of my colleagues in our department. I forgot how long those plots were in place, but at least, I want to say at least 12 years they'd been no-till. We found that no-till soil decomposed tea bags faster. I think we have microbial biomass from those too. That under the no-till, that microbial biomass was also higher.
So kind of simulating these more diverse rotations after corn and soybeans, could you get similar microbiology compared to the oat hay rotation with a more diverse cover crop mixture?
Yeah, that's a great question. It's an area I'd love the government to fund me to do some research. I have some colleagues in Pennsylvania. I think Mark has done some work. I don't know how much soil work he's done with it, but Mark Licht has done some work on cover crop mixtures. I've been mostly my work here at Iowa State has been mostly on focusing on rye cover crops, since that's the most common. But the very few research I have seen on cover crop mixtures, that diversity does have a big benefit. I think there's a plateau though. You know, I hear people putting out these like 32 seed mixtures. I think when you're getting, you know, past a few kind of key plants that provide some of these soil ecosystem services, you start to lose that benefit of the diversity. So in other words, there's kind of a threshold or a cap to the number of species that seems beneficial.
All right, and then this is our last question. If fields have higher biomass, would there be less need for herbicides and pesticides as well?
Yeah, that's a great question. It's an area of research I haven't really gotten into. I'm more on the nutrient and fertility side, but I have seen a little bit of that research that seems like, you know, especially with the diversified rotations, there's big reductions in weed pressure. So herbicide can get cut down. And this could be added to one of those financial benefits we talked about earlier of soil health promoting practices that are almost kind of ancillary or in addition to the benefits of soil health with some of these practices.
So I just wanted to share Marshall's contact information. If you have additional questions or are looking for specifics on any of the projects that he mentioned, he can share with you those research reports. You can follow him on Twitter here. If you did join us for a CCA continuing education credit, please email me by 5 o'clock today with your information there. Sorry about that. And I'll get that submitted to the folks that track your credits for you. So then join us next week. That would be June 25th at 1 o'clock, same time, same place. We've got another great virtual field day with our long-term cereal rye project. So we hope you can join us then as well. So thank you all very much and have a great afternoon.
Thanks for the great discussion and inviting me. This was a lot of fun. I appreciate it.